Okay, so I would like to say that this is the last week of our lectures because this is already week 9 and we have week 10 for your presentations. So the current plan is that we will have you do the short presentation for your final projects next Wednesday. So we will try to reserve a long time slot for all the presentations together. So I think what we will do is that after today's class, we will send you a Google form to fill out your groups. So we will count how many groups are there so we can allocate the time accordingly. So basically how much time each group has will depend on the number of groups. I would say roughly between 5 to 10 minutes. So that's about the time. So I would say that given the short time for the presentation, it's impossible to go over details. And so the key is to present your question, what question you want to investigate from the data, and a short, brief outline of your approach. And if you have some findings already, the preliminary findings, but of course, there's some time between the presentation and the due date of the report. So you will have time to refine your results. But the goal is to present your ideas to me and to the whole class so I can give you some timely feedbacks so you may incorporate into your report. So I will consider this project as a training process for our PhD students in particular, right? This is like a short research project. Given the limited amount of time, you can only explore a certain amount of things, but it will be interesting to try some novel idea, even though the result might be negative, but that doesn't matter. I think the attempt is more important. So we wouldn't say that we only accept positive results. That's unrealistic. And also, I would say use your expertise, whether computational or biological. I think both are acceptable. So you can try to just do something interesting. And I've already seen some interesting projects already. So, and also, of course, if you can combine with your research by adding some data you like to analyze together, that would be encouraged. No, just Wednesday. So we will get everybody, so to give everybody a fair chance. So yeah, so you have the same time for preparation. So we'll put all the presentations on Wednesday. Yeah.
Will there be another homework assigned?
Yeah, exactly. So I would say that the last homework will be assigned this week, likely after the Wednesday lecture, because after I cover all the things, that you will still have 2 weeks to do the homework. So that is independent of the final project. And also I have to admit that this year I covered probably less materials than I did in the past few years. Probably I think I try to teach more basics. And I would say the last part of my lecture in previous years would be like MCMC, try to do how to sample from a posterior. So I'll talk about MCMC in particular, the Metropolis-Hastings algorithm. But then this year, I think I don't have time for that. But also on the other hand, I feel like that's probably not so maybe a little technical and not so useful for everybody at the current stage, because you will only need MCMC when you want to do Bayesian modeling yourself. And that's one option for doing bioinformatics methods. However, that is very specific to Bayesian modeling, I have to say, compared to everything else we talked about, which is more general, more widely used. So yeah, so I think it's okay that I didn't talk about the technical details for that part this year, but I will try to give you the high level understanding about what it is actually about in the last lecture on Wednesday. So that's my plan. All right, so for today, I will just wrap up what I want to say about dimension reduction. So recall that we have talked about PCA, okay? That's what we started. And then we talk about the MDS for preserving the input distance matrix. Then we talk about the t-SNE for preserving density converted from, I would say, the Euclidean distance. You use a kernel to convert the distance to a density, and then you want to preserve that density in the low dimensional space. That's the basic idea of t-SNE. And then we talk about ICA, which talks about independence between the factors the hidden factors you want to find. So because of this independence assumption, we will need to use probability. So probabilistic modeling is needed for ICA, but it's not necessary for PCA. PCA is more like an algorithm. You just use the eigen decomposition, basically. That's PCA. So the idea of ICA, we gave the motivation last time, and we said that ultimately you will convert the problem into a likelihood MLE, maximum likelihood estimation problem. Okay, so that's the high-level introduction of ICA. So finally, I want to introduce another widely used technique, which actually is very interesting because it can bridge dimension reduction with clustering. And this is called a non-negative matrix factorization. And also, by the way, so for the MCMC, I will not talk in detail today. If you're interested out of your own curiosity, you can watch the recordings from my last year. So I think the last 2 lectures from last year were about that. Okay, so this NMF was actually introduced, I think, about 1999. And it was actually in a Nature paper. You know how difficult it is to get a paper like an algorithm paper published in Nature right nowadays. But that Nature paper actually introduced this non-negative matrix factorization technique in terms of doing image analysis. So in that case, basically the application is that, let me still try to use the matrix notation. So this is the matrix notation in NMF. So let me still try to use our old dimension notation for consistency. So I call this V n by P, n observations in P dimensions. And let me say that this W is n by R less than P, and the H is R by P, okay? You can set R much less than P. So in the original application, we would have n images. Okay, let me write it. So in the 1999 Nature paper, okay, so we have n images like faces, facial images, and the P is equal to D squared. So the image, each image is D by D. So D pixel by D pixel. So D rows, D columns, that's an image. So the number of pixels is D squared. Okay, so the goal is to find a representation of the n images in a much lower dimension, R dimensions, and you would have each low dimension corresponds to some combination of the original P pixels. So in other words, in each row of H, it will be what's similar to our loading vector in our PCA, right? So each row is what are the composition, what is the composition of each new feature in terms of the original P pixels. So what I find interesting is that the interesting finding is that each row of H, okay, corresponds to a meaningful subset of pixels. For example, like one row of H may correspond to like the eyes of each person, like the eyes region, and another set of pixels may correspond to the nose, and etc. So therefore, you can see that this gives you very nice interpretation of each new dimension, because each new dimension is a meaningful combination of the original features. And this interesting finding is thanks to the sparsity of W and H. These two matrices have a lot of zeros and only few non-zeros. And the reason is because of the non-negativity. So this is a result of the non-negativity requirement. So basically we require, so if I write my W entries as Wij, okay, and the H entries as Hij, and of course the input V as Vij. So if I just write the three matrices element-wise, then the requirement for this algorithm is that Vij, Wij, Hij are all non-negative. So of course, the range of I and J depends on the dimensions of each matrix, but we're just requiring all the elements in the three matrices are non-negative. So as you can see intuitively without using math, the sparsity of W and H is because we want to make the W and H close to V. So that's called a reconstruction error. We want that error to be very, very small. So the objective is simply the, what is called that? So the L2 norm applied to matrix is called a Frobenius norm. So basically we are going to denote this by V minus WH. So this is the Frobenius norm denoted by F. We want to minimize this, okay, or minimize the square of this norm. So if we write it out, then it's just the sum of the element-wise reconstruction error. So this is equal to, so V, of course, is M by P, right? So we can write that I from 1 to M, J from 1 to P. And we want the difference between the Vij, okay, corresponding to the reconstruction from W and H. So in that case, I can write the reconstruction as, let me say, K from 1 to R, then Wik times Hkj. So that's the reconstruction from the matrix multiplication by WH. I want this error to be small, so that this can guarantee that W times H will give me roughly V by this optimization, but subject to the constraint that all the things must be non-negative. So in this minimization, so this Wik, Hkj, all of them must be non-negative. So that's the constraint. And this non-negativity constraint would introduce sparsity in the solution. What is special of using an AF norm? So it's just when you do least square in linear regression. So this is just like a matrix generalization of least square. So the square difference is the commonly used loss in most optimization problems. And I would say there are two reasons. One reason is that this is just directly a generalization of the Euclidean distance, the most commonly used distance in any geometry, right? That's one reason. And another reason is that this is a very nice function for optimization. You can easily take derivative, right? So I think that's the two reasons for explaining why we use this norm. For optimization simplicity and also for the intuition, which is about Euclidean distance. Okay, so yeah, so solving this objective function under the constraint of non-negativity, you can introduce sparsity. And I would say an intuition is that if, like under the PCA, you can allow the two matrices, both like the, you know, the loading matrix and also the, okay, I'll talk about that later. So this is too abstract for now. So that allowing for the negative coefficients can actually have some cancellation effects. So therefore, the positive ones and the negative ones all add up together can make this approximation very well. But here you don't allow for negative coefficients or negative values for cancellation. Therefore, you have to push a lot of things to zero. And I will say that's the intuition behind why negativity introduced sparsity without using math to say it. Okay, and so related to this, another technique is called non-negative least squares. That is also something that can introduce sparsity in the coefficients. For example, when you run linear regression, you force your coefficients to be non-negative. Then you can introduce sparsity there. Okay, so for solving this objective function, exactly we use non-negative least square alternatingly. So basically you start with some initialization of W, you solve for the corresponding H. Given that H, you solve for W, so you do that update alternatingly. So that's just one common way of doing the non-negative matrix factorization. Okay, so let me just quickly put that. So this can be solved by alternating non-negative least squares. This is often short as NNLS. Okay, so then the interpretation of this non-negative matrix factorization can be twofold. So one is that you can consider two interpretations. So one is that you can consider W, which is M by R, as low dimensional. So here low dim means R dimensional embeddings of the N observations. That's one interpretation, okay? Similar as what you do by PCA. When you do PCA, recall that you could also obtain the PCs, right? So let me just try to do the recall of PCA. Okay, so let's say that here we start from X. Okay, maybe let me just do V to be the same, V and by P. Okay, so what we do by PCA, just as a recall, is that we do eigen decomposition. So two ways are okay. Eigen decomposition of this V transpose V. Okay, we assume that this V has column centered already. So columns, each column V is centered. So in that case, if let's say that we just denote this by, I used the V already, that's the problem. So because in the eigen decomposition, we often use the V as the matrix. Let me see what else I can do. Okay, so let me just do, say, R times this lambda, which is the diagonal matrix, times R transpose. Okay, that's one way to do it. Another way is to do the SVD on the V directly. Okay, so if we do the SVD on V directly, we will write V as, maybe I'll just write the notation, L times also a diagonal matrix. So here, this diagonal matrix would just be, okay, the half of that, which is the singular values instead of the eigenvalues, times R transpose. So either way, you can get the decomposition, and we will care about this R transpose. So basically, let me say that the R transpose is just equal to, okay, so this is P by P. So let me just say that the R transpose is just R by P, which means that you only keep the top R. Okay, it's keep the top R, right singular vectors, which are exactly the same as the top R eigenvectors. So it's the top R right singular vectors of V, or the top R eigenvectors of V transpose V. Okay, so either way, we would have this R transpose, and then given that, you would have the PCs. The PCs would define as V times R transpose. So you see, R transpose is, sorry, my bad. This is P by R. P by R is the R transpose. So with this, you are going, no, my bad. Sorry, I think I messed up with the notation. So R is R by P. Yeah, and then I'm going to do V times R. So this is N by P times P by R. So this gives me an N by R matrix. That is the R dimensional embeddings. So these are the R dimensional embeddings of the N observations. So this is what we do by PCA. So we can still get the R dimensional embeddings just by working on the matrices. And we can see that this dimension V by R is exactly matched with the dimension of W. So W and V times R are both the embedding matrix. But what's different here is that you can see in the PCA, if you multiply this PC matrix by R transpose again, okay, this is the embedding. If I multiply it by R transpose, which is R by P, so this is R by P, then I will restore an N by P matrix. So this will be approximately equal to V. V is N by P, but I lose something. What I lost is by dropping from the eigendecomposition or the SVD, the rest of the right singular vectors or the rest of the eigenvectors. So I only keep the top R as a way for denoising, because I think the top R eigenvectors or right singular vectors preserve the meaningful variance. So for the rest, I drop them. So that's why they wouldn't be exactly equal to each other. But let's say that if I just keep everything, I should be able to restore this V exactly. So I just did rotation. I didn't do any removal of the original dimensions. So you can see that PC has this nice property. But now if you compare this to the NMF, so in the NMF, so in contrast, in NMF, what we try to do is that this W, which is N by R, H, which is R by P, okay? We also want them to approximate V. So therefore, you see the dimensions are completely matched. And then if I regard V as an alternative to VR, and is H an alternative to R transpose? So that's the question we may wonder. Okay, in other words, basically what we want to say is that can we use H as R transpose and take its transpose as R? So maybe what I want to say is that you see, so the question is, R offers a projection of a p-dimensional observation to R-dimensional. That's exactly what we do PCA for. So let's say for a new observation, say for in this notation, say we have a new V star, like a row in V, we say it's in p-dimensional, then we can do V star times let me see, V star is p-dimensional. Then what we can do is that if we want to make it R-dimensional, then we can do either, this is already a column vector, so RP. So yeah, so okay, let's do this. So I can do R transpose times V star, like let's do this way. So this will give me a R-dimensional vector. Okay, so you could do this, or if you put V star as a row vector, just like a row in V, or if I do V star transpose, so this is 1 by P. If you do this, then you could do V star transpose times R, and this will give you a 1 by R vector, row vector. So either way you could do it. So this R offers a projection. But the question is, Does the H offers a similar projection? Does H transpose offer a projection? Right, because H has the dimension exactly as R transpose. Then R offers projection. I would wonder, does H transpose that corresponds to R offers a projection? So if that's the case, then we would need something similar to here. So we know that by the definition, go back, of the eigenvector or singular vector, we would have R transpose times R equals to an identity matrix. So for here, I know what we have is R transpose times R is equal to an identity matrix of R dimensional. This is not a problem. And also for R times R transpose, it's not exactly a p-dimensional identity matrix because we keep the R less than P. So it's not full rank. So what we have is just like a few ones, like R ones here, the rest are 0. So we will have R ones in the diagonal. So matrix like this. So that's why I'm saying that this is just an approximation because you cannot fully restore the identity matrix. So that's the things we have here. But for the H transpose, we don't have this requirement in the NMF. So this requirement would require that. So basically H is R by P, right? So this would require H times H transpose equals to identity R. But this is not a condition in NMF. So in other words, in PCA, we would have the embedding matrix as VR, the loading matrix as R transpose. That's the PCA thing. In the NMF, we just allow W and H to be any two non-negative matrices without requiring any orthogonality condition on the H transpose. So therefore, naturally, NMF doesn't offer a projection. So that's what I put here. So the conclusion here is that the default H hat from NMF is not a projection matrix. Okay, because if it is yes, then we could rewrite. Otherwise, we could write W, the embedding matrix here, by the analogy to PCA, just as V times H transpose. Okay, we could write this, and then we would have the objective function or loss function is this new thing, V minus V times H transpose times H, Frobenius norm squared. So in other words, we don't need W and H as two matrices to find. We just need one H because W can be just written in this way, right? So because this is just a projection of V by the H transpose, that's the embedding. So we change the objective function if we add this new requirement that H transpose offers a projection. And this is motivated by PCA, where we just need to find this R matrix alone. And this R can be found by eigen decomposition. So motivated by PCA, we can change to this by adding the constraint that the elements of H are still non-negative. Okay, and this modification actually has a name. So this is called the projection non-negative matrix factorization. So projection NMF or you can write it in short as PNMF. So it's a modification of NMF by adding the constraint that H transpose must be a projection matrix. While in the original NMF, there's no such constraint. So you can imagine that by adding this constraint, we just reduce the possible search space for H, right? Because that there's more requirement on it. So this is actually this.
Sorry, I have a question.
Yes.
Okay, so yeah, yes, it's not a projection, right? But I mean, like the definition for projection here is a bit confusing because like original data is in a real number space, but the transform data is still in some latent space, but it's not a real number, it's just non-negative. I forgot the notation for that, but it's like in that kind of space. So I guess, is it not a projection into like a completely different like space, in the sense that like it's not real, so it is a projection, but like I don't know how to explain this, but it's
so the PCA is real because PCA has non-negative constraints. So we are using the PCA as a motivation for modifying the NMF. But in the NMF, all the discussion about NMF, all the elements are non-negative. So this has nothing to do with adding a constraint on H or not. So in the original NMF, we require all the elements of W and H to be non-negative. In the new PNMF, what we just added is this new requirement that H times H transpose is equal to identity R, our dimensional identity matrix. That's the only thing we added. But still, we didn't drop the non-negative constraint. So therefore, you see, by calling it projection, we are saying that this is the projection result. So my right multiplying H transpose to V. So V is M by P. H transpose is P by R. So W is M by R. So we just project every P dimensional observation, which is from the non-negative space, to an R dimensional observation, which is still non-negative.
I see. Okay.
Yeah.
Thank you.
Yeah, but in the PCA, there's no non-negativity. I would just say PCA is the motivation here. We just realized that in PCA, we just need one matrix. That's the projection matrix. But in the original NMF, we look for two matrices. And then we realized that if we are motivated by PCA and we want to just find one matrix that serves as the projection, then we can modify the NMF by adding this constraint. I hope that makes sense.
Yep.
Okay.
Thank you.
Okay, thank you for the question. And then you see with this, then this H matrix will be nice, right? So V times H transpose serves the purpose for doing embedding. And H itself has this nice orthogonality condition or property. Okay, so I want to say that this projection NMF is actually something we used in our recent publication for single cell analysis. So we use the property of the sparsity of NMF, which is inherited from NMF, and it still holds for PNMF. So the resulting H matrix will be very sparse. So the loading matrix is very sparse. So in this case, you can see the H matrix, which is R by P. So by being very sparse, we can say what? So for each row, just as in the 1999 Nature paper, we can see which original features have non-zero loadings there. So for those with non-zero loadings, then we can see, like in single cell data, the features, P features are P genes. Then we can call this a gene group, right? And these genes may be functionally related. So then we basically found R gene groups, or some people may want to call them modules. Okay, that's one thing. Another thing is that this orthogonality condition would actually make my R rows pretty much orthogonal to each other. And this is not something that holds for the regular NMF. There's no requirement about the orthogonality, about the R new dimensions. While in the PCA, we naturally have that orthogonality, right? All the PCs are loadings are orthogonal to each other. So here, this orthogonality ensures that the R groups are orthogonal. So therefore, since the loading made the values are non-negative, you don't have cancellation. So therefore, if a gene has a positive loading here, the gene is unlikely to have a positive loading in another row. Because otherwise, if you do the inner product of the two rows, you are not going to have zero. So this can ensure that the R groups are kind of exclusive. And this exclusiveness would make the interpretation very nice because different groups correspond to different genes. Okay, so you can better explain the result. And furthermore, if you look at this embedding matrix, so furthermore, if you look at the embedding matrix, which is V times H transpose, so this matrix also has the explanation. So this is M by R, the embedding. Also using the regular NMF notation, we can call it W, right? W is the embedding matrix. Either way, so this one, because of sparsity, it also can be regarded as R clusters of the N observations. R clusters of the N observations. So this is the second interpretation of NMF. Remember here I said two interpretations. So one is the embedding, dimension reduction. The second is this is the second. So I call this the second interpretation. The second interpretation is clusters. So NMF can also be regarded as a clustering technique thanks to its sparsity. So in other words, here you are going to look at each column of this W matrix to see which observations has non-zero values in that column. So therefore, each column is one cluster. Okay, but here the clusters are not totally exclusive, as you can imagine, unlike the k-means. We don't have that exclusive requirement for that. So even though here for when you do the H transpose, right? So the H transpose is what? Is P by R. So here in each column, of H is actually our loading, and the columns of H are exclusive, kind of. But when you multiply V, that is no longer guaranteed that the resulting W would have exclusive columns. So basically what I'm going to say is that you can use NMF as a way to find new features which are sparse combinations of the original P features. So you have R new features. And also with each feature, you can cluster your N observations for each feature. So that offers a very nice interpretation, both in terms of the original P features, as well as in terms of the N observations. Okay, and this is very suitable for our sequencing data analysis because if we just summarize the read counts, the counts are guaranteed to be non-negative, right? Okay. So if you are interested in this, in knowing more about this, I'm going to post the paper I mentioned from my group, the single cell PNMF. So that contains more details about the interpretation, and you can read that for more understanding, for more detail. Okay, so I think that concludes what I want to say about dimension reduction. So only the NMF or the PNMF require the input matrix to be non-negative, but the previous ones don't have that requirement.
Sorry, I have one final question. Yeah, I know a lot of questions. most of the genetic data, single cell genotype matrices, all of these are non-negative, right? But it's like pretty standard to use PCA decomposition when doing any sort of preliminary analysis. Like, under what cases is it better to actually use non-negative matrix factorization or the projection version of it as opposed to PCA?
Okay, that's a very good question. So I would like to say that people, when they use PCA, the values being non-negative is not really a concern, because as we said, the standard way to do PCA is you're going to do centering. So once you do centering to make every column, every feature have mean zero for all the n observations, you would naturally introduce negative values into your matrix for decomposition already, right? Since the centering requires the column sum to be zero. So you would have positive values, negative values, even though your original values may be non-negative. So that's for PCA. And also, under what conditions PCA preferred? I would say there is a very important result, theoretical result called the Eckhart-Young theorem. So this is the theoretical foundation for doing PCA, basically. So this one says that if you want to do this decomposition, just to minimize this norm without adding any constraint on W and H, then the optimal solution is PCA. PCA gives the best, the solution. So PCA gives you the minimum reconstruction error. And this result guarantees that if you just want to preserve the original matrix as much as possible without worrying about interpretability, sparsity, PCA is the way to go. So that's why, back to your question, why is PCA so commonly used? The reason is that if you have this optimality for preserving the variance, and the PCs are orthogonal, the new features, I should say, are orthogonal, so not correlated. And that's actually very ideal, right? And also you can plot your data in whatever low dimensions you want, like 2 dimensions, 3 dimensions, that can give you a lot of information. And that's just for dimension reduction. So that's why in single cell, actually that's what I want to say. So a data processing step used in many analysis pipelines is to run PCA as the first, I would say the second step. Yeah, so I'll talk about that later. So the first step is people usually do feature screening. Because say you have 20,000 genes from human, but you believe that many genes are not informative. For example, those genes have too many zeros in too many cells, so barely expressed. Then you don't want to keep those genes in your data. So that's why the first step is usually feature screening. You maybe just keep the top 2,000, 3,000 genes that has the largest variance, for example, or has the smallest zero proportion. These are just commonly used criteria for doing a first step screening. Then with those genes, you still have 2,000 or 3,000, then people maybe do PCA to remove that, to reduce that number to say 20 or 30 PCs. And then you start from there. So basically your feature matrix become the number of cells by 20 PCs. And you go from there, you do any downstream analysis. So I would say in those cases, both the feature screening as well as the PCA can be regarded as denoising steps. You just want to retain the majority of variance you believe that belong to biology, and you want to remove the other variance that could due to technical noise. And also if like 20 PCs can preserve a large proportion of variance, you are happy. Since your variance is well preserved, but your feature number is so much smaller, like from 2,000 to 20. So therefore, you have less worry about the dimension. This is called the curse of dimensionality. So when you have a large dimension, a lot of things become different. So that's why people don't like high dimensions. And the PC is a very effective way for reducing dimension, but still retaining a small reconstruction error. So you preserve most of the variance, and then you do other analysis. So that's very different from what people do negative NMF, non-negative matrix factorization for. So here, the key is more about interpretation. So because your original P features have meanings, right? Then if you have the sparse loading, then for each new feature, you can say which original features correspond to it. And I have this nice interpretation. Well, for PCA, you may not. Even though you can talk about the loading absolute values, but it's not as clear cut as like 0, non-zero. You have to set a threshold on the loadings in PCA to say, oh, these are the features that contribute most to my first factor, second factor, and so on. So you have to say that. While in NMF, you just have a natural result. So I will say PNMF offers a very nice intermediate between NMF and PCA. So kind of borrows the advantages from both, but its optimization is as not straightforward as PCA. So like for NMF and PNMF, there's no guarantee that you will have a unique solution unless you have some theoretical conditions. But for PCA, the uniqueness of the solution is very good, and you have a very fast algorithm because you just need to do SVD, basically. So I think that all can explain why the PCA is so popular. So maybe I said too much, but the one sentence summary is that if you care about interpretation, I would say go for NMF or PNMF. But if you just want to reduce dimensions but preserving the variance in your data, go for PCA.
I see. That's helpful. Thank you.
Okay, thank you for the question. Do people have other questions before we move on? Okay, so yeah, that's actually what I want to say about the last part. So it's about the practical issues in dimension reduction. And I already said both, but let me just try to summarize them again. So one is we want to do feature filtering often before we run dimension reduction. And this is also some steps. This is also needed before clustering, I should say, before dimension reduction and clustering. Yeah, so needed for both. The feature filtering is that you want to filter out features that are uninformative. You don't want them to affect your analysis. So remove uninformative features. So one criteria many people use is the so-called coefficient of variation. So which is defined as the standard deviation over the mean. So this is under the idea that for many distributions, the variance may increase with the mean. Okay, so like for example, Poisson, we know that the larger the mean, the larger the variance, right? So therefore, people say if I just compare the variance, that may not be so meaningful. So the variance comparison is only meaningful if I consider the mean difference, and that amounts to the definition of the coefficient of variation. So many people rank features by the CV, and they just keep the top features with the largest CV. So this is one approach. Keep the top features with the largest CV. Okay, CV means coefficient of variation. That's one approach. And for single cell data, because it's super sparse, you may also consider the zero proportion, or I would say proportion of non-zeros, yeah. So here, you still want to keep the top features with the largest percent of non-zeros. So that's commonly used for single cell data, because if a feature just has all the zeros or 99% of zeros, it's barely useful. So you don't want to keep them. So these are the feature filtering. This step can remove a certain amount of features, so that cannot that I would say, relieve your curse of dimensionality. And the second thing you may consider is data transformation. And this is also related to the question about the non-negativity, actually. Yeah, so I would say this is still an open question. When you have a count data matrix, would you want to work with it as is, or would you want to do some transformation before working on it? And I think that depends on the technique you use. The PCA, actually, if you know the probability interpretation, it's called the probabilistic PCA. It basically assumes normal distribution or Gaussian distribution. And also, if you think about Euclidean distance, it makes more sense when you have data that's kind of spherical-like in a circle or in a bowl. That's kind of data when it works well, and also k-means clustering. So therefore, for a lot of cases, you do want your data to kind of like normally distributed. So those classical techniques developed for normal data would be applicable. So thinking of that, many people commonly apply this transformation, which is called log count plus 1. This is a very common transformation applied in the single cell field, and you can see the effect. The effect is that for the count distribution like Poisson or negative binomial, you know that they are heavily right skewed, right? So like what I mean is that for Poisson, this is like count, and this is like a probability. So if you have that histogram for those cases, often for like Poisson or MB, negative binomial, for many common parameters of those two distributions, the distribution looks like something like this. So you may have a long right tail, which means that you have some large counts with not too small probabilities. You may get those large counts. And clearly this distribution looks not normal, right? So you have this long right tail. But the log transformation, you know, when you apply it to large positive values, it will shrink those values towards the center. So therefore, this distribution effectively can make the distribution more bell-shaped like. Okay, so even though you are still truncated at 0, since a count of 0 will give you 0 after log transformation. So that's why we need to have this plus 1. Otherwise, log 0 will be negative infinity. You want to avoid that. But still, even though you have this truncation at 0, okay, so I'm saying that if you plug log count plus 1, you still have this 0. But now the probability, this distribution may be more bell-shaped. Okay, so you may have something more like this, and this shape will make normal distribution-based methods more applicable. So that's why many people do this before they run PCA. And you will see that this is actually the recommended strategy in the popular single-cell pipeline like Surat or ScanPy. Okay, that's the first transformation. Another transformation I have seen people do in genetics is called the inverse normal transformation. This is what we have seen, similar to what we have seen in the quantile normalization. So in other words, you just convert your data to ranks, and then you want to find, or you normalize the ranks into quantiles, data quantiles in your data, and then you would apply the inverse normal transformation into the values, and those values will follow a standard normal distribution. So to be exact, let me say that you have n numbers, okay? You have n numbers, observations, maybe say n observations, one-dimensional, okay, in one dimension. And let me call them x1 to xn, okay? And you first obtain the ranks. So the smallest one will receive a rank of 1. The largest one will receive a rank of n. So let me call them R1 to Rn. Okay, so basically I would say Ri is equal to 1 if xi is the minimum, and Ri equals to n if xi is the maximum. Okay, so that's the way we define the ranks. And then what you can do is that you can convert the ranks to quantiles. So basically quantiles must be, oh, I should say not quantiles. Oh, no, I think that's bad to say. Not yet quantiles, I should say normalization. Normalize the ranks, normalize. So normalize, I mean, I just want to make the ranks between 0 and 1, not exactly equal to 0 or 1, because the quantile of 0 in the normal distribution equal to negative infinity, and the quantile of 1 equal to positive infinity. I want to avoid that. So therefore, you see for the smallest rank, it's not 0, it's 1, that's okay. But for the largest rank, if I divide it simply by n, I would get a 1. And when I convert 1 into the quantile from normal, I will have that positive infinity problem. So therefore, here what we do is that we will divide each by n plus 1. Okay. So once we do that, we will go get those percentages in terms of the ranks. And then finally, we would convert them by inverse normal CDF. So get the quantiles of standard normal. Okay. And let's denote the phi, this big phi function, to be the CDF of standard normal. So the quantile is defined as the inverse of the CDF. So basically, I will have the inverse of each one to the inverse of this. So by inverse, what we mean is essentially, given this is the standard normal density, okay, mean 0, standard deviation 1. So if the left tail here is x, then the value here is the quantile of x, which is the CDF inverse at x. That's the definition. So as you can see, we get the quantiles as values from this normal curve. Then, of course, these n values would follow standard normal. So in other words, this inverse normal transformation preserves the ranks, like the smallest remains the smallest, the largest remains the largest. The ranks are preserved, but the values are altered. We alter the values to be from the normal distribution, and what we preserve is the rank. Does that make sense? So therefore, the new data, these data will be what you work on, and they would follow standard normal distribution, even though your original data are not. I think in genetics, like the GTech consortium for the EQTL analysis, then in that case, they're doing a linear regression. Like the Y response variables are a gene's expression. The response variable is one gene's expression, and we have its measurements in different individual samples. We also have those individual measure for the SNPs genotype. So one SNP is our predictor. So we get that SNP's genotype 0, 1, 2 in those individuals. And then we run a simple linear regression. So in that case, the Y gene expression, we're undergoing this inverse normal transformation. And I guess the reason is that they want to use a simple linear model where the assumption is that the error terms follow a normal distribution. So that's why doing this transformation will make the linear regression apply better. And otherwise, if the Y's do not follow like a normal distribution, you have to worry about like the whether you should do the generalized linear model instead of a linear model. So that's my understanding of why they did this. So I would say that data transformation is something applied statistics often concerns about, because sometimes you don't have to stick with a model. Sometimes you don't have to develop a model specific for your data. For example, if you believe that you want to apply some well-defined or popular method, but somehow your data just doesn't satisfy the assumption about the data distribution in that method. One way is to modify your data by running a monotone transformation. Like here, the log transformation and this inverse normal transformation are both monotone. You preserve the ranks, that you can make the distribution satisfy that method assumption, so then you can use the method. This is one approach. And the other approach is that you can develop a method for your data without altering your data. So those two perspectives, I would say there's no consensus about which one is better. So my own perspective is that it depends on your expertise. So for example, if you're a method developer, you may prefer to work with raw data and come up with a model that can explain your data. But if you're a method user, you want to apply a popular method, but unfortunately, by checking the assumption of the method, you found that your data don't satisfy the assumption. Then using monotone transformation may be a reasonable way so that the method can apply better. So that's my own perspective about these two approaches. But of course, we can keep thinking about this. It's about case by case. Okay. So that's all I want to say about dimension reduction and clustering. So we concluded the unsupervised learning part mostly. Yeah, Chris, do you have a question?
Yeah, I have a question about data transformation. And so when you do either the log or the inverse normal transformation, are you doing that for each feature?
Yeah, exactly. You're doing that for each feature. That's a very good question. So I think for the count one, you don't have any, you apply it to the whole matrix element wise. Yeah. And for this one, you do it for each feature. But one thing you definitely alter is the correlation structure.
I was going to say that, like, if you do that, you're kind of assuming that there may be some independence or something between the, you change the correlation a little bit.
Yeah,
right?
But we didn't assume independence, because you can see within each feature, the ranks of the n observations are still preserved, right? So, but the thing is that the correlation on the original scale could be different from correlation on the normal scale. Yeah.
Okay, thanks.
That's exactly right. Yeah. Okay, thank you. Yeah, but I want to say that so in the eQTL study genetics, the reason I think they can do that is because when they run the regression for one gene versus one SNP, they are doing that just separately for every gene. They are not using the gene-gene correlation. Okay, so that's it. We concluded the unsupervised learning, which I think we spend the most time on in this class. And so the next topic I really want to talk about is about variable relationship, which is actually following from the question about correlations. So this is a new topic. Let me see my number. Okay. Okay, so this is about, I call this measures of association. So I wouldn't actually call this as unsupervised learning or supervised learning, because this is not a topic that's in a standard machine learning textbook, right? Since in this case, we are not really saying about n observations, what we do about them, even though we can use the n observations to help us estimate those associations. So I would say this is more like an inference problem. Yeah, it belongs to, if you still recall, we spent some time talking about the distinction between statistical inference and what we say, the machine learning, the learning problem. So the learning problems concerning about the observations and observations, while the inference is concerning about the population structure. In here, the measures of association is about the association between two variables on the population level. That's the thing we want to infer. Okay, so in this case, let's say that we have two variables, or more generally, two random quantities. So let's say that we have x. Okay, let me just make it simple. Let's say x is a p-dimensional random vector. Y is a q-dimensional random vector. Okay, so p and q can be integers. So we start from the simple thing. So first we start with p equals q equals 1. Both are scalars. That's what we start. And we have two variables we concern about, and that's the population structure. But in our data, maybe I put here. So in the data, we have n observations for these variables, and we put them as x1, y1 to xn, yn. So n observations about these two random variables measured in pairs. So that gave us the data for doing the inference. So we are going to talk about several measures of association. And the first one, the simplest one, is linear association. And for this one, it's the Pearson correlation. Okay, so the Pearson correlation is the most commonly used thing for two variables. So on the population level, let me just give this notation between two variables x and y. It's defined based on the joint distribution of those two variables. Both are one-dimensional, and it's defined as the covariance of x and y divided by the square root of the product of variance of x times the variance of y. Okay, and this definition, actually, this is ensured by what we call the Cauchy-Schwarz inequality. That can guarantee that the square of the covariance is less or equal than the product of the two variances. So that will give us that the Pearson correlation lies in the interval from -1 to +1. So that's the population quantity we want to estimate. And for any joint distribution, we can define this quantity specifically to the bivariate normal distribution. So in that special case, so if we have this xy, okay, follows a bivariate normal. So in this case, we can write the normal distribution as the mean. I can put mu x, mu y. And the variance here is a 2x2 matrix. So that would actually be the variance of x, the variance of y, the covariance of x and y. Okay, so in this case, we actually have a very special property. So in this special case, not all the joint distributions are bivariate normal, but when it is, we would have that the covariance x and y equals to 0. You can see this is equivalent to saying the Pearson correlation of x and y equal to 0. And this is equivalent to saying that x and y are independent. So we use this perpendicular sign to mean independence. However, this is only specific to bivariate normal. If they're not bivariate normal, then in general, having the Pearson correlation equal to 0 doesn't imply independence. But in general, rho x, y equal to 0 does not imply x independent of y. Okay, so this linear association can be calculated, or estimated by the data, right? So to estimate this rho xy. We use our data, and that's what we call the sample Pearson correlation. So we can call this maybe using a hat, a standard way to call an estimator. Can be just calculated. This estimator can be calculated by estimating the covariance and the variance respectively. So we know that the sample covariance based on the data has this interesting formula, 1 over n minus 1, I from 1 to n, xi minus x bar. X bar is the average of x1 to xn. Yi minus y bar. Okay, that's the sample covariance. And for each of the sample variance, we can use the variance formula. So that's I from 1 to n, xi minus x bar square. Okay, that's how we estimate variance of x. And to estimate variance of y, yi minus y bar square. Okay, and then you can see the n minus 1 can be canceled. So what we are left with are just the cross product and also the square root there. So this is the formula for the Pearson correlation we use in practice. Xi minus x bar square, yi minus y bar square. Okay, that's it. So Pearson correlation is the most commonly used strength for measuring a linear relationship. And one more thing I want to say is that you can see it's symmetric, right? So this is symmetric between x and y. So it doesn't matter which one you put in the front, and its range is from -1 to 1. So many cases people use its square if they don't care about the sign. They just use the square of it and they call it r square. So r square x and y is the square of the row hat. Okay, and this belongs to interval 0 to 1. So you don't have negative values if you use r square. And also this has another interpretation if you run linear regression. Okay, so this is equal to the r square of linear regression. That is if you run the y1 to ym, you regress it on x1 to xn by linear regression, by simple linear regression. This is a very interesting thing because when you do simple linear regression, it's not symmetric. So like the line you obtain by regressing y on x is not equal to the line when you regress x on y. So these two lines are not equivalent of each other. So basically here in this r square, it's defined as this thing. It's defined as yi hat, the fitted yi minus y bar square over the original yi minus y bar square. So this is the r square formula. And the linear regression guarantees that the average of yi hat equals to the average of yi. So that's why they share the same y bar. So this is the property by linear regression. So that's why they share the same y bar. Okay, and because of symmetry, right? So this is also equal to the r square of running the reverse regression. If you regress x1 to xn on y1 to ym. So I think this is something very interesting. So even though you don't have the same line, don't have symmetry in linear regression, but somehow they have the same r square. So you can check it in R, and that's a very interesting relationship. And they are both the square of Pearson correlation. Okay, so I think I will just stop here for the Pearson correlation, the most commonly used relationship for measures of association. And next time, I think I will try to wrap up the remaining of the correlations I want to talk about. So if needed, so depending on you guys, maybe I will do a vote on the next class to see if you want me to go one more lecture next week or not. It's totally up to you. And then we will wrap up, and also we will look forward to your presentations on next Wednesday. So I think we will send a Google form for everybody to fill out about your groups, so that then we can allocate the time and send the presentation order, which will be random. Okay, so I will see you next, I'll see you on Wednesday. Yeah.
Do you have like 5 minutes for a quick question on the project?
Sure, you can just stay in the Zoom.
Okay, let me know when.